Okay, so this will be the second half of the urinary system discussion. Today is the second lecture on this material, and I just want to quickly pick up with a review. Before I do this, though, let me remind you that this material, the urinary system, along with the acid-base regulation that I'll mention at the very end of this lecture, plus the reproductive systems that I'll discuss next Tuesday, will be the content for your final lecture exam. That'll be a full-scale 100-point exam. That'll be next Thursday. Next Tuesday, don't forget, you have your lab exam. That'll be from 2, either at 2 or at 7. And that lab exam will also be in the blue and gold room. And I'm holding it there since I'll have extra large numbers of students at the lab. We'll have that lab exam in the blue and gold room, again, either at 2 or 7. Don't forget that you also have your Connect assignments to do. You have your 2 quizzes, one on urinary, one on reproductive systems. At the end of this presentation, you can certainly go ahead and get started on the urinary quiz. And there are still some extra credit quizzes out there if you haven't already done those. So use your time wisely this weekend and come in as prepared as possible for the lab exam, and then followed by the lecture exam next Thursday. So last time, I just want to ramp back up into this. Last time we discussed the regulation of the GFR, that is the glomerular filtration rate, how much filtrate is being produced by the kidneys. And we discussed how the GFR is considerably stable across a very broad range of blood pressures, that within those blood pressures, the GFR is kept constant, and it's kept constant by 3 different homeostatic mechanisms. The first is considered autoregulation, sympathetic control is the second, think nervous system, and thirdly, hormonal control. So just a real quick recap. We did this last time. So renal autoregulation is actually broken down into 2 ways, one myogenic mechanism and the tubuloglomerular feedback. The myogenic mechanism, remember that smooth muscle, when it is stretched, fights back, it constricts back. And so as there is more blood going through the arterioles, in the afferent arterioles, that increased blood pressure will cause the smooth muscles to simply fight back, if you will, and actually then reduce the amount of blood coming into the glomerulus. That, of course, would then change how much blood is going in, and then as the pressure drops, the afferent arteriole would once again relax. And so you have this constant fluctuation going on, which is going to keep the filtration rate rather stable. There's also tubuloglomerular feedback, and this is the mechanism by which, as it sounds like, the tubes, that is the loop of Henle, is going to come back and speak to or inform or control the glomerulus. This is happening at a portion of the renal corpuscle referred to as the juxtaglomerular apparatus, and this is at the very end of the nephron loop. After the nephron loop has come back up and re-entered into the renal cortex, the nephron loop will come in contact with the afferent and efferent arterioles right at the vascular pole, that is at the blood end of the renal corpuscle. In this juxtalar glomerular apparatus, in the JGA, there are 3 special kinds of cells. Number 1, within the nephron loop. So we're in the nephron loop, we're inside the nephron loop, right where the loop comes in contact with the afferent and efferent arterioles. And there's a patch, a spot, a stain, if you will, a cluster of cells called the macula densa. Those cells are especially sensitive to changes in flow and in fluid concentration. And when they are activated, they're going to secrete a paracrine. That is a locally acting molecule, which will then stimulate the second type of cells, the JG cells, or the juxtaglomerular cells. These are basically large, smooth muscle cells that will be in the afferent arteriole, and they're directly across from, very close to, the macula densa. When they are stimulated by the macula densa, they are going to either dilate or constrict based upon what signal is being sent. So that's going to continue to control the fluid. So if there's more fluid in the nephron loop, then the kidney, the nephron would say, whoa, this is too much fluid. And so it's going to send a signal back to the glomerulus saying, hey, we've got enough fluid right now, restrict the amount of fluid or the amount of blood coming into the glomerulus. Remember too that the JG cells are the renin supplying cells. So that renin, that enzyme, is released in response to low blood pressure. So really these JG cells are doing two things. They're responding to the macula densa, and they're responding to changes in blood pressure by releasing renin. Finally, there's the mesangial cells. Mesangial cells, as the name suggests, mes, middle, angio, vessels. So the mesangial cells are right in the middle between the afferent and efferent arterioles and scatter up between the capillaries of the glomerulus. And they too are going to communicate through paracrine signals with the macula densa and the JG cells, and they're going to either constrict or relax the capillary flow. Again, what we're doing is overall, we're reducing or regulating the amount of blood that's coming into the glomerulus, therefore regulating the amount of the GFR. So this boxed area right here, this area would be referred to as the JGA, the juxtaglomerular apparatus. And this is all review. But again, here's the nephron loop. The cells within the nephron loop are the macula densa. The JG cells are wrapped around the afferent arteriole, and the mesangial cells are right there in between, as well as scattered between the capillaries of the glomerulus. So that was all the autoregulation. And then in addition to that, there is the ability of the sympathetic nervous system to have quite an effect on kidney and on the GFR. That is, when you are in a sympathetic mode and there's epinephrine and norepinephrine being released, that is going to cause the constriction of the afferent arterioles. Again, this is going to reduce the GFR, reduce urine output, and of course during sympathetic surges, less blood is going to your kidneys so that more blood can be directed toward your heart, your brain, and your skeletal muscles. Finally, there is the hormonal control, and that is where that renin comes in. Remember, the renin is supplied by those JG cells if the blood pressure drops. And what they're going to do, if there's a drop in blood pressure, then the kidney will release renin. The liver will always be producing this protein called angiotensinogen. So it's already floating in the blood. The renin will cleave that angiotensinogen into a smaller molecule called angiotensin 1. That molecule doesn't do a whole lot on its own. Instead, it will undergo conversion by this enzyme ACE, supplied by the lungs, to make angiotensin 2. Now, angiotensin 2 is a very potent vasodilator. Vasoconstrictor. So what it's going to do, what angiotensin 2 is going to do, is speak to the hypothalamus and tell it to release more ADH. That's going to cause you to be thirsty and elevate your blood pressure. It'll also cause vasoconstriction of your vessels. Again, if you squeeze in the vessels, it'll raise your blood pressure. Thirdly, it will send a signal to the adrenal cortex to release aldosterone. Aldosterone will be released and will now cause sodium and water retention. Again, all of these mechanisms are going to lead to a correction in or restoration of the blood pressure. So that's all about filtration, if you will, right? Controlling the filtration process that's happening at the glomerulus. In addition, though, we want to think about the stages of urine formation. Again, this is review. So the stages are as follows. First, you have filtration, right? And that happens in the glomerulus. Then there is both reabsorption and secretion. That's happening along the length of the proximal and distal convoluted tubules, as well as the loop of Henle. And then at the end, the kidneys are going to be conserving water by removing water from the urine and concentrating the wastes so that your the urine is as concentrated as possible. And we'll spend some time in a minute understanding how is it that that water is pulled out of the urine, and how is it that the urine becomes concentrated. So we talked about the PCT. Last time I told you that it's difficult to see it under the microscope, but if you were to look at proximal convoluted tubules, you would be able to see very prominent microvilli, as well as an abundant number of ATP, or of ATP producing mitochondria. Again, microvilli to increase the surface area, very long length, again, to increase the surface area, as well as a lot of ATP production, because many of these reabsorptive processes are actually active transport mechanisms. In fact, you use a pretty significant chunk of your resting ATP and calorie consumption simply on the reabsorptive process. So as I was finishing up last time, I told you that the molecules that are reabsorbed are going to be reabsorbed through one of two mechanisms. Either these molecules are going to travel through the cell. This is going to have the greatest control. So things that are moving through the cell transcellularly will be welcomed into the cell, usually through a receptor, and then transported through the cell to the other side. Other molecules will simply go around the cells paracellularly, and they're going to go through leaky junctions in between the cells. The things that move through the cells, out of the tube, or between the cells are eventually all going to be taken up by the peritubular capillaries. Remember, those peritubular capillaries are wrapped around the distal and proximal convoluted tubules. Now recall, where do we find sodium? Sodium is going to be found where? It's going to be the number one electrolyte in the extracellular fluid. So there's a lot of sodium in your plasma, and therefore there'd also be a lot of sodium in the filtrate. And so we're going to see that sodium is going to be the key to moving molecules. Sodium is going to create an osmotic, think concentration gradient, as well as an electrical gradient, because remember, sodium is positively charged. So it's going to create a gradient, two types of gradients. And as molecules like glucose and chlorine ions and other chloride ions, as other potassium ions, acid, as all of these things are reabsorbed, we're going to see that sodium is the key molecule. Many of the molecules will be moved out of the tube into the blood through symporters. Symporters are basically protein transporters, but they're unique. These require sodium to travel along with the other molecule. So the way that your body is going to gather the glucose and many of the amino acids is actually to have a sodium also travel with it. There are also antiporters, that is, transporters where sodium is going one direction and acid is going in the other direction, hydrogen. There's also, don't forget, if we weren't careful here, we could have sodium accumulating too much in the cells. And remember that sodium-potassium pump that we referred to back in the action potential chapters is constantly working. And that's going to make sure that sodium does stay out in the extracellular fluid, right? So we refer to this as secondary active transport. So yes, there is ATP going on to run the sodium-potassium pump. And when sodium is moving through these symport mechanisms, it doesn't directly consume ATP. So we say that it's secondary active transport. Again, wherever sodium goes, chloride ions will follow, and that's more of an electrical attraction. Remember, chlorine is negatively charged, whereas sodium is positively charged. So they're going to attract each other electrically. So not to get too bogged down with this, but let's take a look at this image. So here we are in the tubule on the right-hand side. So within the tubule, right, we've just brought fluid from the glomerulus, and we're in the proximal convoluted tubule, and there's a lot of sodium in here. But not only is there a lot of sodium, there's also a lot of glucose and potassium and chlorine and all these other molecules that are important to maintain, that you don't want to lose. You want to recycle and reclaim these or reabsorb these molecules. And what you see here is that many molecules are going to go straight between the cells. This is called the paracellular, and it's going to be picked up right here by the peritubular capillaries. Other molecules are going to have to go through these symport. So as you see, as glucose comes in, sodium comes in with it. As chlorine Sorry, and then here's the antiporter. When sodium comes in, hydrogen goes out, acid goes out. There's also a number of aquaporins. Aquaporins, as the name suggests, right? IN, a protein that creates a pore through which water passes. So aquaporins are protein channels for water specifically. And so there's a lot of water also being pulled back into. And then as the molecules come into the cell, they also have to be transported out of the cell. Now, all these little red circles remind you that all of these processes are actually active transport mechanisms. So there's energy involved. And like I said, there's a lot of energy necessary to keep these levels properly balanced. So glucose is transported, again, out of the tube into the blood through a transporter. And we'll talk about this in a moment when we talk about the glucose reuptake in diabetics. But the sodium glucose transporter, you need to have sodium in order for glucose to come into the body. Now, this is an interesting fact. Urea is also reabsorbed. Now, that may seem counterintuitive to what I mentioned in the first lecture. I told you that urea was one of the nitrogenous wastes, that your body is filtering this out of your blood, and that you would expect, I think, that urea would simply continue its journey into the urine and be released from the body. But in fact, about 50% of the urea will actually be reabsorbed and recycled. Now, other nitrogenous wastes like creatinine, it does not get reabsorbed at all. And we're going to see that this urea that is reabsorbed is going to be important in maintaining the salinity of the medulla. So when we get to that moment in that conversation toward the end of this, and I'm talking about the salty marsh or the salty medulla, keep in mind urea is going to help contribute to that process. Now, why do we have to reabsorb? What's the practical problem here? Right? We only have 5 liters of blood. We only have about 3 liters of plasma. Yet the kidneys every day are producing, in a male, about 180 liters of filtrate, in the female, about 150 liters. But of that 150 to 180 liters, the person's only going to release 1 or 2 liters of urine a day, which means that the vast majority of the urine of the water and of the filtrate must be reabsorbed. Now, when I say reabsorption, I want you thinking about the proximal convoluted tubule. There's a tremendous amount of reabsorption here. That's why there were so many microvilli in the proximal convoluted tubules. It's here that 2/3 of the water, about 65% or so, of the water will be reabsorbed. There will also be a large number of the solutes, the sodium, the glucose, and the salts also taken up here. And again, the water is taken up by these aquaporins. And the water that is taken up in the proximal convoluted tubule is referred to as the obligatory water reabsorption. This is not under hormonal control. So I'm going to write hormone here and then draw a line through it. There's no hormones involved in regulating this water uptake. This is obligatory. In other words, it must happen. The kidneys are going to reabsorb this water regardless of hormone conditions in the body. Now, again, when these molecules, the water and these solute molecules, are reabsorbed, as I've shown you, they're going to be reabsorbed into those peritubular capillaries, which are wrapped around, right? Those peritubular capillaries are wrapped around the proximal and distal convoluted tubules. Now, what is it that's going to promote the movement of the water into the capillaries? Recall, right? Things don't just move. Things move because there's either a concentration gradient or there's a pressure gradient, but there's always something responsible, or maybe a solvent, right? Solutes suck. There's always going to be something that is going to drive or drag molecules from one area into another. Well, one of them is an accumulation of reabsorbed fluid around the cells will create a really higher interstitial fluid pressure, and that's going to drive water into the capillaries. Secondly, narrowness of the efferent arterioles. I want you to think about that for a second. The efferent arterioles are the outdoor, aren't they? The outward door for the glomerulus. So if you have a narrowing of the efferent arterioles, that would lower the blood hydrostatic pressure in the peritubular capillaries. And that would create less resistance to reabsorption. And then finally, if there are proteins in the blood, which there are, of course, that's going to elevate the colloid osmotic pressure, and that's back to the idea that solutes suck. So if there's a higher COP, then this is going to drive more COP. If there's a high COP, that's going to drive more of the water into the blood. So what I'm trying to show you is that many of these molecules that are being reabsorbed are moving through transporters. In other words, there's a specific protein in the proximal convoluted tubule through which molecules must travel. And there's a limited number of these transport proteins. If the transporters are being completely occupied, if every single transporter is occupied, then there is this idea of saturability. There's only so many transporters available, and if they're all being used, then any of those solute molecules beyond the maximum would then not be reabsorbed and instead would show up in the urine. This is classically illustrated with glucose. So a person who has a normal metabolism and what we would call normal glycemia, right? Normal levels of sugar in the emia in the blood. This is a simplified glomerulus, and this is a simplified Bowman's capsule. And you know there's glucose in your blood, so glucose, of course, will be filtered into the proximal convoluted tubule. But we want to reclaim the glucose. We want to reabsorb it. And so there's going to be the opportunity for glucose to be taken up out of the tubule. Now, what this figure is not showing is that what is out here? Right out here, there's going to be peritubular capillaries, and so that glucose would be taken up back into the bloodstream through the peritubular capillaries, and as such, there would be no glucose in the urine. That's normal. In a diabetic, however, there's already such an elevated glucose in the blood that there's so much glucose being filtered that no matter how much the transporters try, they can never capture all of the glucose, so there will always be some glucose showing up in the urine. Again, we would call this glucoseuria, and you've seen this in the lab. From a blood standpoint, if the blood glucose level, right, in the blood gets above 220 millimeters or milligrams per deciliter, then that's going to result in sugar, quote, spilling over into the urine. So a diabetic, well, first of all, a normal blood sugar is going to be something in the high, 80, 90, 100. Even after a sugary meal, it might go up to 110, 120, 150. But the body quickly reabsorbs that sugar. In a diabetic, that number stays artificially high. And so they will start seeing glucose spilling over into their urine. Now, there's also the process of secretion going on. So not only is there reabsorption, there's secretion. And secretion is the opposite. It is the process by which the tubule is going to extract or take molecules directly from the blood and put them right back into the tube. Okay? So secretion, very, very important, especially for acid. Now, secretion is also important, though, when you think about drug clearance. Why do you have to take aspirin every 4 hours? Why do you have to get penicillin 3 times a day? Why do you have to continue to get more morphine if you're on a morphine pump? Well, it's because those medications, those molecules, are quickly cleared or broken down and filtered out of your body. And we call this the rate of clearance. Now, some medications are not cleared very quickly, so they hang around in the blood longer. And so, you don't have to worry about that. Those molecules, or those medications wouldn't have to be given as often. And as we'll see, secretion is critical to acid-base balance. So holding on or regulating your hydrogen as well as your bicarb as far as buffering is absolutely critical, and we'll be discussing that at the end. So what's the deal with the nephron loop? I'm going to spend the next few minutes talking about the nephron loop and why it's so important. Well, there's really two kinds of nephron loops if you think about the two different kinds of nephrons. There are cortical nephrons, and as I've described, cortical nephrons have a very wimpy, very short loop of Henle. There are, however, 15% of your nephrons called juxtamedullary nephrons. And those nephrons have very, very long loops of Henle that dive deep down into the medullary pyramids. And that's where I'm talking about right here. The primary function of the nephron loops, especially the ones that are juxtamedullary, is to generate a salinity gradient, a salty medulla. And you want a real salty medulla because your collecting ducts are traveling right down through that saltiness of the medulla. And as the urine is traveling down the salty medulla, you're going to see that water is going to be drawn out, and that's going to allow us to concentrate the urine and effectively, very effectively, conserve water for our body. There's also some reabsorption occurring in the nephron loop. So when you hear about reabsorption, it's happening in the proximal and the loop and the distal tubule. But the primary amount of it's happening in the proximal convoluted tubule. Now, if you go back and look at the numbers, you're going to see that the fluid that arrives in the distal convoluted tubule, that is, it's already been through the proximal, it's already been through the loop of Henle, is going to still contain between 10 and 20% of the water, and still quite a bit of the salts. Well, that seems great. I mean, yay, we've already reduced it by 80 to 90%. However, if we still urinated all of that, you would be urinating as much as 36 liters per day. So we still need to work on concentrating the urine. It has to be concentrated significantly more. Remember, we're only going to release 1 to 2 liters a day. Now, the release of water is going to be constitutive or obligatory back in the proximal convoluted tubule. But by the time we get to the distal convoluted tubule and to the collecting ducts, there are going to be a number of hormones that are critical to the regulation of water. I'm going to list them and then go through each one. Aldosterone, atrionatriuretic peptide or ANP, ADH, antidiuretic hormone, and parathyroid hormone also has quite a bit to do with salts, especially calcium salts. Now it turns out that in the distal convoluted tubule and in the collecting ducts there are two kinds of cells. You're not going to be able to tell them apart, but the most numerous cell, the principal cell, are in fact called that same thing. They are the principal cells. They are the most numerous. They have the receptors for the hormones. They are the ones involved with water and salt balance. The remainder of the cells are called intercalated cells, right? They're stuck in between, if you will, intercalated cells, and these are the cells that are involved with the secretion of acid. So let me go through these with you. Aldosterone or aldosterone is the salt retaining hormone. We've already talked about aldosterone in the hormonal control. I mentioned to you already that if there's a drop in blood pressure, that the JG cells will release renin. The renin will then form angiotensin II ultimately, and all of this is going to also stimulate aldosterone from the adrenal cortex. However, in addition to that, this steroid will also be released when sodium concentrations fall. Well, what am I telling you? Aldosterone is a salt-retaining hormone. So the aldosterone is going to cause your body to retain salt, to hold on to salt. Well, that makes sense that if your sodium levels are starting to drop, your body would want to retain more sodium. Again, we have to keep in mind how critical sodium is. It's critical to the function of your kidney. It's critical to the ability of your cells to undergo action potentials. It's critical for everything. So we've got to keep our sodium levels well controlled. It's also true that when sodium levels fall, potassium levels rise, right? They're typically found in inverse relationships. So aldosterone will be released when sodium levels are low, potassium levels are high, or there's a drop in blood pressure when renin is released from the JG cells. I've already said that here, but what happens is that the aldosterone, when it comes to acting on the water reabsorption, is going to work on the thick segment of the nephron loop. When you look at the nephron loop, you're going to see there's a thicker section, and then there's a thinner section. And so the thicker is called the thick segment. And here, what's going to happen is that there's going to be a reabsorption of more sodium and a secretion of more potassium. And you know that water and chlorine always follow the sodium. And so basically, if you reabsorb more sodium, you will also retain more water in the body. And this is going to help you keep your blood volumes and blood pressures intact. Of course, while you're having aldosterone released and you're conserving water, your urine output would be reduced. And as I said, there's secretion of potassium going on, which means that there'll be more potassium in your urine. Second hormone, atrial natriuretic peptide. You'll also just see it called the atrial natriuretic hormone. So you may occasionally see it just referred to as ANH. Now this hormone is going to be released from the atrial myocardial cell. So the heart is also talking to the kidneys. If the blood pressure is too high in the atria, then these cells in the atria sense it and will release atrial natriuretic peptide. Now what this is going to do, atrial natriuretic peptide is going to work on the kidneys to, of course, fix the blood pressure problem. So it's ultimately going to reduce the blood volume and the blood pressure. How is it going to do that? 4 different ways. That ANP is going to cause the afferent arteriole to dilate and constrict the efferent arteriole. Well, if you open the door going in and shut down the door leaving the glomerulus, then that would increase the GFR. Increased GFR, increased water, right? Let go, reduce pressure. ANP is also going to inhibit renin. It's going to inhibit aldosterone, and it's going to inhibit ADH. So again, ANP is going to do everything it can to cause urination. So it's going to inhibit renin, aldosterone, and ADH. Finally, it's going to inhibit the sodium chloride reabsorption by the collecting ducts. Again, if you're inhibiting reabsorption, that means that more sodium is going to be flushed and water will follow solute, and so you'll have more urine output. ADH, we've been talking about this since the very first lecture. ADH is produced by the hypothalamus, stored and released from the posterior pituitary. And recall that there are osmoreceptors in the hypothalamus. And so if you have if you're dehydrated, then you're going to have a rising blood osmolarity. Those osmoreceptors will detect that and stimulate the hypothalamus to release more or to synthesize more, and for the posterior pituitary to release that ADH. What that ADH is going to do is make the collecting ducts more permeable. Another way of thinking about it is going to cause more uptake, more reabsorption of the water. And this is going to happen in the distal convoluted tubules and in the collection ducts. Finally, there's parathyroid hormone. Recall we talked about this back with skeletal homeostasis, calcium homeostasis, but PTH is secreted from the parathyroid glands whenever there's a deficiency in calcium. And what this is going to do is when there's low calcium, obviously you want to increase those calcium levels. But remember, if you're going to increase your calcium, you also need to, if you're going to increase calcium, you also need to decrease phosphate. So, in order to increase calcium, we're going to increase phosphate excretion, get rid of more phosphate. And ultimately what this is going to do is, because the phosphate's not being retained, the calcium ion will stay around a little bit longer. This is going to be important for overall bone health. And again, we see that a hormone here is regulating the distal convoluted tubule and is related to reabsorption, specifically here of calcium. So as a quick review, of this whole process. The proximal convoluted tubule is going to reabsorb about 2/3 of all of the filtrate, the water and the solutes, and that's going to be returned to the peritubular capillaries. The nephron loop is going to reabsorb another 25%. So that means that by the time we get to the distal convoluted tubule, we've already taken, these numbers would add up to 90%, right? So you've already taken away 90% of the water. But like I said earlier, 10% is still too much. So in the distal convoluted tubule, we're going to continue to reabsorb water and sodium and chloride under hormonal control, especially by aldosterone and ANP. We're also going to see in the distal convoluted tubule secretion specifically of hydrogen, think acid, and potassium, think the opposite of sodium. The changes that occur in the urine in the distal convoluted tubule, that represents the end of the changes to the chemical composition of the urine. But even once we have changed all the chemical composition, we still have the opportunity to conserve more water in the collecting duct. So the job of the collecting duct really is to, and the distal convoluted tubule, is to do that last very critical job of the kidney, and that is to conserve water. Right? We have to get rid of metabolic waste, but we also have got to conserve water. We cannot have excessive water loss from the body. So the kidneys are working constantly to concentrate your urine so that we lose as little water as possible. Take a look at this simplified glomerulus. Right, so here's the glomerulus, here's the tubes, here's the proximal tubule, down the loop of Henle, back up distal tubules and into the collecting duct. Cortex above, medulla below. This is the collecting duct going right down the center. Remember, you're in the middle of one of those pyramids. And I already told you, and I'll say it again, that as you go down in the medulla, it gets saltier and saltier. The osmolarity goes from 300 all the way down to 1200. Just think of it as getting really, really salty. And as the urine is traveling down the collecting ducts, that high salt is going to draw the water out. And as a result, your urine is going to become extremely concentrated. Right? So we need to have this saltiness in the medulla in order for your urine to become concentrated. Critical, critical, critical. As the urine passes through the increasingly salty medulla, the water will leave and the urine will become more concentrated. So we need to control, right, how much water our body loses. Diuresis, right, is urination, if you will. So if you drink a lot of water, and some of you did that in lab this week, and you saw what happened to your urine. If you drink a lot of water, you're going to have a larger volume of urine, and it will be rather hypotonic, meaning there'll be fewer solutes in it. Another way of saying that, you've all learned that if you have lower solutes, that also is going to lower your what? Specific gravity. As we lose the water, though, the salt is staying back in the urine. Okay? If you are dehydrated, what's happening to your urine? We all saw this. Your urine becomes hypertonic, has more solutes mixed in it, and your urine is more concentrated. Well, if your urine is more concentrated, your blood osmolarity will also be going to stimulate the posterior pituitary to release ADH. The ADH will then cause more aquaporins to be inserted into the tubules. More water will be reabsorbed, and the water will be removed, and the urine will become more concentrated. So if a blood pressure is low in a dehydrated person, the GFR will also be quite low. Now, how does this happen? What I'm trying to scream right now is that the medulla of the kidney must be salty. And it must be salty in order for the urine going through the collecting ducts to be concentrated. It turns out that the medulla will be about 4 times saltier than the cortex. And this is going to work through nephron loops because there is what's called a counter current multiplier. That is, the medulla is constantly recapturing salt and returning it to the medulla. And there's always a flow of fluid in the counter current or in the opposite direction through the tubules. Let me show this to you in a moment. As fluid is flowing down the descending limb, so the loop of Henle is described as having a descending limb and then an ascending limb. So as things are coming down the descending limb, it's going to be passing through a saltier and saltier environment. It turns out that the descending limb is very permeable to water. That means that water can leave, but it is not permeable to salts. So as it's going down, the fluid itself is becoming very salty itself, and it will get to the bottom to be very, very salty, about 1,200 milliosmoles at the bottom. Now that unit milliosmoles may not mean much to you, but just know that it's very, very salty. Now on the way back up in the ascending limb, it turns out that the cells of the ascending limb do not have aquaporins. They are impermeable to water, but they are very permeable to the electrolytes. And what this is going to do, and you'll understand it more in a moment, is it's going to maintain a really high salty or high osmolarity of the medulla. There's also, remember I told you there was urea being reabsorbed, and that urea is actually contributing to much of the saltiness in the deep medullary tissues. Constantly, we're recycling urea and concentrating it in the medulla to keep this saltiness in place. Look at this simplified nephron. Actually, this isn't even the whole nephron. This is just the proximal convoluted tubule dumping down into the loop of Henle and coming back up out the distal convoluted tubule. And like I said, on the way down, you're going through the medulla. It's getting saltier and saltier out here, and water is being pulled out. Now, at first you might think, well, wait a second. If water is being released into the medulla, wouldn't that also be diluting the saltiness of the medulla? And yes, it would. But what this picture does not show is that right next to this loop of Henle, remember there are going to be capillaries like the vasa recta. And so whatever water is released out into the medulla will then be taken up into the vasa recta, so that this water is not sticking around here. But what is going to stick around here is that on the ascending side, as we come back up, the kidney will be releasing a lot of the ions. And those ions, again, are going to stay out in this medulla. Right? The medulla is from about this point down. And so all these solutes, all these electrolytes are going to be adding to the saltiness or to the salinity of the medulla. So again, the vasa recta, you've seen that term. The vasa recta is that capillary bed, right? Vessels that are straight, vasa recta, and it's coming down off the efferent arteriole, traveling around the juxtamedullary nephrons. And again, we always see these pictures in isolation, but there's always going to be these capillaries right next to the tubules. The capillaries also have a lot of exchange for salt for water. So again, they're going to be, keeping the water out and keeping the saltiness in the medulla. So let me show you this picture, although I'm still not happy with this picture because I think it really still is confusing. Here's your vasa recta. Now the vasa recta is not sitting over here all by itself on the right hand side. Instead, that vasa recta, pick it up and put it basically right here. Right? It's right on top of it. It's right next to this nephron loop. So imagine that it's right over there. And like I said, in the downward slope, what's happening? As we're going down, water is being pulled out. Okay, so water's coming out. But again, where's that water going? It's coming right into the blood vessels, into the capillaries. And then on the way back up, you've got solutes being put out into the medulla. And there's also urea being put out here. Remember I told you there's a lot of recycling of urea, and that urea is also being exchanged back and forth. Bottom line is it is definitely very, very salty in the medulla. The deeper you go, the saltier it becomes. Also note that those little red circles tell you that this is an active transport process. Remember, active transport requires energy, and the reason it requires energy is because the molecule would not want to go to a more concentrated place. Diffusion is always moving from a higher concentration to a lower concentration. But what we're doing here is we're actively shoving salt into the medulla. Well, whenever things move up their gradient, it requires energy. So the body is spending a lot of energy to keep the medulla salty so that it can concentrate the urine. Again, this is more of a review idea, but what's going on here? This is showing, again, the whole glomerulus. Let's get the whole idea here. So coming in, it's bigger. There's your afferent arteriole. Comes into the glomerulus, filtration occurs, going out the efferent arteriole. Filtrate will start going down the proximal convoluted tubule. Leaving the proximal convoluted tubule and being reabsorbed into the peritubular capillaries will be all these molecules, right? These are all those small molecules, glucose, amino acids, proteins, vitamins, all of that stuff, sodium, magnesium, chloride, you mentioned it, water, all of that stuff is being reabsorbed. So the blue box represents reabsorption. Also, though, in the proximal convoluted tubule, there is some secretion. And secretion, remember, is the movement of molecules from the blood directly into the tube, and our waste products move this way. Urea, uric acid, creatinine, and acid. And some drugs like penicillin, morphine, are secreted directly from the blood into the tubules and end up in the urine in high concentrations. Going down now the nephron loop, this is where we are concentrating the solutes so that we had a salty medulla. And then coming back up, the distal convoluted tubule is also still involved with reabsorption. Nowhere near as much. Sodium, water, and bicarb largely under hormonal control. Right, so there was no hormones over here, but there are hormones on the distal side. And there's also secretion, again, acid and potassium, things that I've already mentioned. And then as this urine travels down the collecting duct, we see that water and urea are being removed. The water is helping to concentrate the urine, and the urea is being removed again, and it's going out into the medulla to increase that osmolarity. So that's what's going on in the kidney. That's sort of a quick overview of the function of each part of the nephron loop. The last little bit that I want to go over, and this is what you did in lab, is just urine analysis. And you know that you can take your urine, you can look at its color. We could have even been really personal and done odor of your urine in lab. We didn't do that, but you can imagine, you may all know this, that freshly released urine is not that smelly. But urine that's been around a little bit longer, because of the bacteria will degrade the urea, which is a part of our urine, and convert it to ammonia. Now ammonia stinks a whole lot more. And so that's like an old diaper, right? A baby pees into a diaper, it's not a big deal. A couple of hours later, that diaper's getting pretty ripe, right? That's why we have diaper pails with covers on them. So we know that the urea gets converted to ammonia through bacteria through time. There's also pyuria, that would be the presence of pus in the urine, or hematuria, the presence of blood in the urine. Now, those wouldn't be normal. Pus in the urine because you have an infection, hematuria because perhaps you have a UTI, you've had trauma, damage to the kidneys, or you have kidney stones that are irritating the mucosal layer. So this is what we're doing in lab. Specific gravity, remember this is a 1.000 would be pure water. And so anything that's dissolved in your urine is going to drive your specific gravity up. pH range, again, you're going to see different ranges. I apologize because there are so many different sources for this, but I think in the lab I said the normal range was 3, I think I said 4 to 7 in lab. Here I'm saying 4, 5, up to 8. But normally, usually, your urine is going to be slightly acidic. So again, think about it. Your kidneys are getting rid of that extra acid. Chemically, your urine is about 95% water and 5% everything else. It is normal to find these molecules. It's normal to find, urea and potassium and creatinine and uric acid and many other molecules. And those molecules can combine into crystals, which you'll see under the microscope. It is not normal to find glucose. Again, indication of diabetes. Hemoglobin, indication of blood or of damage to the kidney. Albumin, proteins in the urine are sometimes indications of hypertension or other issues. Ketones, again, an issue of diabetes perhaps. Bile pigments, maybe the liver's not doing well. So it's not normal to find those things in your urine. Volume, every day, 1 to 2 liters. That's normal. Excessive urination over 2 liters, polyuria. Oligouria would be a decrease or a deficient output less than 500 mils a day. And anuria would be a lack of, essentially a complete lack of. Well, basically, if you have a lack of urine, your kidneys are shut down. Why are they shut down? You've got significant kidney disease. You're so dehydrated that your blood pressure is so low that your kidneys aren't working. You have circulatory shock. Again, shock is always characterized by low blood pressure, not enough blood pressure to keep the kidneys going. Or perhaps there's a blockage. In men, the prostate enlargement could actually block the ability of the urine to pass through the urethra. Less than 400 milliliters a day, and you're in trouble, right? Remember I said 500 milliliters a day was obligatory. That's how much your body really needs to get rid of every day in order to get rid of the waste products. Anything less than 400 or 500 mils a day, and you're really going to start becoming toxic. You're going to start building up these waste products. Diabetes, okay, and I mentioned the transporter earlier. If there's sugar in the urine, but there's actually 4 different kinds of diabetes, and they all affect urine volume. That's really where its name comes from. Diabetes mellitus is characterized with having polyuria, right? You have excessive urination. Well, with that, you also have polydipsia. In other words, you're always thirsty. So you have excessive thirst because you're always urinating. Well, why does a diabetic urinate more? Well, because they have glucoseuria. Because they have glucose in their urine, remember, solutes, water will follow the solutes. And so the solutes are going to suck. So the more glucose in the urine, the more water that will leave in the urine. So again, polyuria. There's another type of diabetes referred to as diabetes insipidus, DI. Now this has nothing to do with glucose at all. DI is a hyposecretion, a complete lack of, or very low levels of ADH. Remember, ADH is an important molecule for regulating your urination levels. If you don't have ADH, then your body can't anti-diurese. It cannot stop or control the urine flow. And so individuals will pass ridiculous amounts of water, right? They're urinating all the time. And again, they have a lot of polyuria and polydipsia as well. So those are the same, but DI has nothing to do with sugar. If you wanted to increase your output, you could take a diuretic. Diuretics do different things. There's different types of diuretics. Some diuretics, like caffeine, will cause a dilation of the afferent arteriole. Some diuretics, like alcohol, will inhibit ADH. Others will work on the loop. And what they're going to do is inhibit some of the electrolytes from moving. Well, if you're going to impair the countercurrent multiplier, that means you're not going to make the medulla as salty. And if the medulla is not as salty, then you won't pull as much water out of it, and therefore your urine would be more copious, right? You'd have larger output because you can't concentrate the urine as much. So if a person has hypertension or congestive heart failure, they would certainly be giving them diuretics, trying to get that fluid off the body. If not one, maybe 2 different diuretics, again, trying to work in multiple ways to keep that water off the body. And then in lab, we did some basic renal function tests. There was much more to this, but we did look for some issues that we could measure through the urine. I mentioned renal clearance. That is, renal clearance is the amount, how quickly a particular molecule is removed from the blood through urination, right? Renal clearance, how quickly something is cleared from the body. Well, what three processes are going to regulate this? Well, if you're going to take something from my blood and put it into my urine, first of all, it has to be filtered. So the amount of filtration, number one. Number two, I'm going to add to that the amount that is secreted. Remember, secreting is adding it to the urine. And then I'm going to take away the amount that was reabsorbed. And when I take the sum of those, I have now my renal clearance. I am not going to discuss this slide, so we can slide over this slide, not to worry about any numbers there. And I'm also not going to mention anything here in the bottom of this, although I will say, if you do have kidney disease, right? If your kidney's not working well, then it would lower your GFR. So if you have a patient with kidney problems, you want to be measuring their medications very carefully. Because if their kidneys aren't working well, that means they're not filtering and they're not getting rid of these metabolites, and they wouldn't be clearing as quickly. So if you kept giving them the same amount of medications as a person with normal kidneys, those medications would start to accumulate. So people with kidney issues have to be very careful on their dosing of medications. The next 10 slides are just a basic review of the bladder. So I'll just quickly go through these. And we have the ureters, of course, taking the kidney down, or they're taking the urine down from the kidney down to the bladder. And the bladder is this muscular sac that's going to have three layers to it. And in the muscle layer of the kidney, in the muscle layer of the bladder, there is the detrusor muscle. And this is a smooth muscle that is going to help to relax and to squeeze out the urine upon urination. If you look into the bladder, there's the trigone. That is that triangular area that is bordered by the opening for the two ureters and for the outgoing urethra. Typically, a full bladder is about half a liter. A really full bladder, 700 to 800. And as the bladder gets distended, it's going to start to fill and expand superiorly within the abdominal pelvic cavity. And the inner layer will thin out. And that's why we say that this is transitional epithelium, because it is going to transition from being thicker to thinner. So here we can see the trigone. Right, the trigone is this area inside, two openings for the ureters coming down, and the outgoing urethra. This would be the internal urethral sphincter and the external urethral sphincter, which is found in the same layer as the urogenital diaphragm. This is female because I don't see any prostate gland, which would be right here if this were male. Okay, so I think I got a little enthusiastic and went over and I've repeated myself a little bit. So I'm going to now merge this file with the file that was taken and create one file that will be uploaded to YouTube for you. I'm going to start recording right here. A few minutes ago, this thing shut down on me, so I'll be going back and redoing that first part later. Okay, so slide 79. Yeah, okay. So what do we have here then? The distal, now we're in the distal convoluted tubule. We just finished up with the hormones. So the distal convoluted tubule and the collecting duct. Let's go back and just think about this. The proximal convoluted tubule, what was it doing? Reabsorbing about 2/3 of all the filtrate, returning it to the peritubular capillaries. Mostly water, lots of other mechanisms, and we said that sodium wasn't critical there. The nephron loop is then going to reabsorb another 25%. So how much is that? How much is that? 65 and 25 is 90, right? So what I'm saying is that most of it's been reabsorbed by the time we get back to the distal convoluted tubule. And then in the distal convoluted tubule is where we're now under hormonal control to collect the rest of the water. And aldosterone and AMP are especially controlling this. Along the way, we're reabsorbing and we're secreting. And by the time we get to the distal convoluted tubule, we've pretty much done all the changing of the urine that we'll ever do, right? It's urine. But what it isn't yet is perhaps as concentrated. So by the time the filtrate goes through the proximal tubule, through the loop of Henle, and through the distal convoluted tubule, all the reabsorption and all the secretion will ever happen has been done. We've already changed the chemical composition of the filtrate. It is now urine. The only thing the distal convoluted tubule can now do, along with the collecting duct, is change the concentration of water. So we can make our urine more or less concentrated, but we are not going to continue to change the chemical composition of the urine. So that brings us finally to this last little bit on water conservation. Okay. We've been talking about this, so we know that these hormones are involved with this. And these hormones are acting where? Distal convoluted tubule or the collecting ducts. And again, it's the kidney's job to make sure that it's keeping our fluid levels as flat as possible and, again, concentrating the urine. Let's take a look here and figure out how the kidney is going to make your urine more concentrated. What do I have here? I've got blood coming in to the glomerulus, and I've got filtrate, and it's coming through the what? Through the proximal convoluted tubule. What is this thing not showing me? Right? This thing is not showing me any of the capillaries that are wrapped around here, aren't they? Right? This is just looking at the tubules. And then down the loop of Henle. See how that's thicker on that side? I'll talk more about it in a moment, but see how that's thicker on the way down? So that's the thick segment. I haven't shown you a picture of that yet. And then see on the way it comes back up around? That's thinner. That'll be the thin segment, and I'll come back to that. And then we get up into the distal convoluted tubule, and then we go into the collecting duct. Okay? Now, the collecting duct is receiving urine from multiple nephrons, isn't it? There's urine coming in from multiple nephron units. And what we're seeing in this picture is that water is being drawn out. All these outward arrows showing water leaving the collecting duct. So what does this mean? The urine is becoming what? More concentrated. What is drawing that water out? Remember I told you that the medulla is very salty, a salinity gradient, or the salty marsh is what I'll keep calling it. And so down over here it says the osmolarity, and man, 1200, that's very salty. And what you see is that as you go deeper in the medulla, as you go deeper down in the medulla, these numbers go from 300 to 1200. So now what is urine? As the urine is coming down the collecting duct, what is it doing? Becoming more and more concentrated. If there wasn't the salty marsh, what would happen? We would be urinating out urine that was way too dilute, right? And we'd be urinating all day, right? But that's exactly what would happen. So we need to have the saltiness of the medulla to help concentrate the urine. We'll figure out in a moment how that medulla becomes so salty. So again, as it says right here, as the urine passes through the increasingly salty medulla, water will leave by osmosis, causing the urine to become more concentrated. It can become 4 times more concentrated through that passageway. How do we control it? Our body is always drinking water. And that water, if we just drink large amounts of water, and some of you have already experienced this in the lab this week, what are you creating? Drinking large volumes of water will produce large volumes of hypotonic urine, suggesting what? Urine with very little stuff in it, right? Very clear urine. And what other word would be used to describe that, those who have been in lab? Very watery urine would have a very low specific gravity, right? We use that term in lab. So very hypotonic urine would have a low specific gravity. And that's okay, right? If we're drinking a lot, we want to get rid of a lot of that water, and we don't want to carry a lot of stuff with it. So it makes sense that if we're increasing a lot of water output, that we're going to have it mostly pure water. But here's what happens. We create, right, hypertonic urine. What does that suggest? Hypertonic means what? Not volume, but tonicity, meaning that there's a lot of, you're dehydrated, right? And there's not as much water, which means that it's very concentrated. Well, let's think about this. If we have high concentration of solutes, what gets stimulated? Right, the osmoreceptors will get stimulated, won't they? And now you'll be putting out ADH, and that will do what? Cause you to retain water. How are you going to retain water? Your kidneys would make more aquaporins, right? Put more aquaporins in the tubules. And now you're going to reabsorb more water in the collecting duct, and your urine would once again become more concentrated. So what we're talking about just the control, right? If you're drinking a lot, you're going to have hypotonic urine. If you're dehydrated, you're going to have hypertonic urine, and your body will produce ADH. What about if your blood pressure is low in a dehydrated person? If blood pressure is low in a dehydrated person, well, if blood pressure is low, what's going to happen? And what would be produced? Low blood pressure going to the kidneys, you would make some renin, right? And if you're already dehydrated as well. And so this is going to definitely decrease or reduce your GFR. So what would happen is that your filtrate's going to go more slowly through the glomerulus. There'll be more time for reabsorption. That means there's more time for salt removal, and less urine would be produced. Right? Lower blood pressure. Think about it. The blood is not being pushed in as much. There's less filtration, but the filtration is moving more slowly. And the slower this stuff moves, the more the chance it has to get reabsorbed, and it would capture more things and reduce the amount of urine that's being produced. Now, how in the world does the kidney make a salty medulla? We know why, right? It has to be a salty medulla to do what? We have to have a salty medulla to concentrate the urine. And we all recognize if we didn't have a concentrated urine, we'd be peeing all day. Right? We've got to concentrate it. Okay. In fact, the medulla is about 4 times saltier than the cortex. To do this, there is a counter current multiplier. So the nephron loop acts as a counter current multiplier. What this means is that the fluid going down and around the nephron loop is moving in the opposite direction of adjacent tubules in the nephron loop. So we've got nephron loops moving in different directions, and there's also, quote, a multiplier. That means that we're constantly recapturing the salt and returning it to the medulla. So just know that the nephron acts as a counter current multiplier. So as fluid flows downward in the descending limb, as fluid goes down, here's what happens. Do you agree that the fluid is passing through an increasingly salty area, going down the loop of Henle? Turns out that most of the descending limb is very permeable to water, but not to sodium chloride. By permeable, what does that mean? That it can get out. So on the way down, we're losing water, but we're not losing salt. Water passes from the tubule into the extracellular fluid. The salt is left behind in the tube. And what this is going to do is highly concentrate the fluid. Does that make sense? If you're in the tube, you're going down the loop of Henle. And if you're a little salt molecule, and you're going down the loop of Henle, and all the water is rushing out, do you agree that you're now more concentrated as you go down the loop of Henle? Now, on the way back up, the ascending limb is impermeable to water. But you can move the ions. And this is where we're going to keep our high osmolarity of the medulla. Okay? I'll show you a picture in a moment. There's also a recycling of urea. Now, we said earlier in the very first lecture, very first slide or so, that urea is a nitrogenous waste. We want to get rid of it. And we do desperately want to get rid of urea. However, urea is also important in creating the salty marsh. So when I say salty marsh, I don't want you thinking just sodium chloride. It is a highly osmotic marsh. So there's a lot of molecules in the medulla, and urea is one of those. And that urea is constantly being cycled through there and helps to maintain that high concentration of salinity or of osmolarity in the medulla. So let's take a look at this picture. Now, what I don't like about this picture or any other picture is it always shows these things in isolation. But let me talk about this, and I'll tell you why I don't like this. So we're going down, and your urine is going from the proximal convoluted tubule and is going down. And as it goes down the tube, what did I tell you? Water can come out. Sodium chloride cannot. So at first that seems counterintuitive, doesn't it? What would be happening there? If I'm losing water, my marsh would be getting less salty. And that doesn't seem right at first. But we have to remember there's an upward swing. So as we're losing water on the downward, when we come back up, here, water can't get out, but what can get out are all of the electrolytes. And that's what's going to really maintain the saltiness down here. Again, it's going from about 300 at the top down to about 1200 at the bottom. Now, what we lose in perspective here is that this nephron loop is not diving down into the medulla by itself. There are other nephrons right next to it. And those other nephrons are doing what? Those other loops are doing what? So there'd be, let me draw it in as a different color. There'd be another loop here. Okay, right next to it. So you would have water coming in and you would have water going out, and you'd have, all this sodium chloride moving back and forth. So I just want you to think about this is not in isolation. You've got densely packed nephron loops next to each other, all interacting with one another. It's not this one loop down into the medulla. What kind of capillary bed is around the loop of Henle if we're dealing with a juxtamedullary nephron? Remember, the juxtamedullary nephrons were the rarer type, only 15%, and they were the ones with the long, deep loop of Henle. And around that was the vasa recta. Well, guess what? Not only do we have, and I'm going to draw this over here, that would be a nephron loop here, but what else is going around it? Don't forget, we also have, right, we have vasa recta coming down through here. So not only do we have fluid coming down here under high pressure, but we also have blood flow. Guess what? That exchange now is going on between the loop of Henle and the vasa recta. And this is what's called the countercurrent system, because you have the movement of blood and the movement of the fluid in opposite directions. And these descending capillaries now allow for the exchange of salt for water. So again, this is going to keep the marsh quite salty. The ascending capillaries exchange salt for water. And again, what this is going to keep doing is it's going to keep giving salt back. So the vasa recta keeps giving salt back to the kidney and results in the increased osmolarity of the medulla. So here's the picture. And this is a little bit better, although it's still simplified. We're looking at this. Now, what you want to almost imagine is that this vasa recta is sitting right on top of this nephron loop. It's not over there on the right-hand side. It is sitting right next to it. When you saw the pictures that I described last time, you know that the vasa recta was wrapped around the tubes. So they're not isolated like this shows. They're sitting on top of each other, essentially. So as urine or as filtrate is going down the loop of Henle, what was coming out? Water. Where's that water? That water is being what? Sucked right up into the vasa recta. Right? So it's leaving the tube and going right back into the bloodstream. And then on the swinging back up on the ascending side, you saw these electrolytes being brought out here. So a moment ago I said it seems kind of counterintuitive, doesn't it, that we're making more water as we go down. As we go down, the water is being added to the marsh. But now we appreciate that the vasa recta is pulling up that water. So what you're still doing is leaving a very salty area. Now there is some salt that can be picked up into the vasa recta. But ultimately what this results in is, again, a very, very salty marsh. And if it weren't for this countercurrent, if it wasn't for the vasa recta going deep down along the nephron loops, we wouldn't have this ability to keep the marsh salty. And we need the salty marsh so that as the water goes through, or as urine goes through the collecting ducts, that it becomes more concentrated. Now, what percentage of the nephrons are involved with keeping this salty marsh? This salty marsh is maintained by the 15% of the juxtamedullary nephrons. The other 85% aren't involved with this. The other 85% are mostly involved with what? What's going on in the peritubular capillaries? That was reabsorption and secretion. Reabsorption and secretion. That's what's happening. 85% of your nephrons are cortical, and they're doing the job of reabsorption and secretion. 15% of your nephrons have this special job, and their special job is to keep the marsh salty. Does that make more sense than maybe it did 10 minutes ago? Lastly, I just want to point out that many of these little you see the electrolytes moving. See the big red circle? That red circle tells us that it's active transport. That is, this is an energy requiring process. So to keep the medulla salty, it requires ATP. This takes work. This is not something that happens naturally. It wouldn't happen without the input of energy. So you're not going to move molecules to where they don't want to go, right? We know from day 1, lecture 1, 105, that molecules move by diffusion down their concentration gradient. What are we doing here? We're concentrating. We're pushing salt where it wouldn't want to go. And that's always going to require energy. So there's a certain amount of energy that's necessary to keep the medulla nice and salty. So this brings us back to the other nephrons. This is primarily what's happening with the proximal convoluted tubule and the distal convoluted tubule. I know this is small, but I'll walk you through this. What sort of molecules are being... Now look at this arrow. This arrow is going from here out. So if it's leaving the proximal convoluted tubule and going into the blood, what process is that? Right? We're reabsorbing. Right? If we leave the tubule and go into the blood. So what sort of things are leaving the tubule and going into the blood? Glucose, right? Glucose, amino acids, proteins, vitamins, all the things we've talked about. Sodium, potassium, calcium, magnesium, chloride, all that stuff, right? Water. What does this arrow represent down here? Going into the tubule. That's secretion, right? So the pink is representing secretion. What sort of things did I tell you were being secreted? Hydrogen, acid. That's the story I want you to know the most. That acid is being directly dropped from your bloodstream into the urine. Some drugs, like I mentioned, aspirin and morphine, are quickly broken down and secreted. And then there's more of your waste products, urea, uric acid, creatinine. Right? Your waste products are so substantial that the body has to secrete them to get them even more into your urine. Over on the distal convoluted tubule, over here, we've pretty much reabsorbed all of the metabolites, but there's still some reabsorption going on of sodium and bicarb. And there's still secretion of acid and ammonia. Now, what kind of nephrons are primarily doing this work? The job of reabsorption and secretion is largely done by the cortical nephrons, right? And the medulla vasa recta story was maintained by the juxtamedullary nephrons. Okay, so I'll go back and I'll re-record that beginning section. I apologize. And that'll all be one lecture. I'll get it all posted tonight. So I'll go back and do that up through the hormone story. Someone recap this for me. What have we learned? Tell me about the overall function of the nephron. Just start popcorning out some ideas. Say it again. 3 types of regulation to keep the GFR steady, right? We talked about autoregulation and hormonal and nervous system. And then when it comes to the nephron, what's the nephron doing? It's filtering the plasma. It is reabsorbing, reclaiming, most of the good stuff. It is secreting, adding stuff to the urine, taking stuff away, and then at the very end, the urine is being concentrated through the collecting ducts. There's 2 different kinds of nephrons. Cortical, primarily with lots of peritubular capillaries wrapped around the DCT and PCT, and that's where most of your reabsorption and secretion's occurring. 15%, though, of your nephrons are of a different type, juxtamedullary, and their anatomy allows them to dive way down deep with their loop of Henle into the medulla. And in doing so, those diving deep loops of Henle do what? Maintain the osmolarity, the saltiness of the medulla. And that happens because of the exchange between the loops of Henle and the vasa recta. And that exchange, because water is permeable on the way down and salt only on the way up, keeps the marsh very, very salty so that the collecting ducts can do their job. We need both types, don't we? We need both types of nephrons to be working fully so that this balancing act, and obviously that 85-15 split has, something to do with the kidney working well. I don't know that there's any evidence that one type of nephron would be destroyed or would break down faster than the other. So I think that the decline of juxtamedullary and cortical nephrons would be the same with age and with disease. Anything in there that's not making sense? I'm sorry, I saw your hand and forgot to say, Des.
Yeah,
What do you got?
That's what the question is. I gotta find it. Okay. Oh, on the functional relation, this one, I don't think it's getting yours, but it's like the water's going out of the capillaries, not that one, the one before it.
Okay, the water is going out of the capillaries. It is. So this is the vasa recta, right, coming down.
Yeah.
Now, what I want you to see, though, look at this really quick. I haven't talked about the numbers too much, but even the blood, as you're traveling down the vasa recta, look at the numbers, 300, 400, 600, 1200 down here. What's that about? Water's leaving. So even the blood is becoming saltier, if you will, as it goes down, right? So the blood's becoming saltier because salt comes in. Okay, salt can come in. So it's like the opposite. Let me just finish this thought and then I'll come back to your question. But as you went down the loop of Henley, what happened? Water went out. And over here, what happened? Water was able to come in from this side, but able to go out on the other side. Okay, so you'll see that the concentration, the osmolarity increases not only in the tube, but it also increases in the blood. Keep going.
Oh, I just thought it was kind of counterproductive, but I think you just kind of said it.
Remember, what I don't like about this picture again is that these things are overlapped right on top of each other, and they're not in isolation. So again, there would be another nephron loop here, another nephron loop here, another nephron loop here. And there would be other vasa recta over here, and they would all be communicating and exchanging one with another. The end result, though, is what? Increased osmolarity as we go deeper into the medulla. And you know that the collecting duct goes right down the center of the medulla, right through the saltiness of the medulla, concentrating your urine along the way. Any water that gets taken out of your collecting ducts will just simply be taken right up into your blood, right up into the vasa recta, to keep that saltiness. It'll sink in. I know it will. Okay. Now, the majority of the rest of this is the things we've been doing in lab. So I'm not going to spend a lot of time here. I'm just going to walk through this and remind you of what we did. In lab, we did urinalysis. And urinalysis allowed us to look at the chemical properties of the urine. We could also have looked at the physical properties, that is the color, the odor. Did anyone analyze odor? I don't think we did that, right? But we could have gotten really, personal and examined the color or the odor of your urine. And we know that you can spin down the urine and look for crystals and casts and cells and particulates. Now, in the lab, if you were doing the 30, 60, and 90-minute urines, there probably was very little odor to the urine because there wasn't much time. But in older urine, your first urine sample might have been more odoriferous, right? Because there are bacteria degrading the urea in your urine to ammonia, right? And this is like, stinky old diapers, right? Diapers aren't so bad right off. But you store one in the back window of the car on a trip, and you know that later on, right, it really stinks. So what's going on? The bacteria that are part of your urine are degrading the urea to ammonia, which is far more nasty smelling than urea itself. So you don't want old diapers, right? You could find the presence of pus in your urine. If you had an infection, you would have pyuria. And some of your urines maybe were cloudier. Maybe you're dealing with a UTI you didn't even know you had. Or you could have hematuria. Typically not a good thing, but if you had a significant UTI and your kidneys were a little bit damaged in the short term, or you had had some trauma to your kidneys, maybe you got a kidney stone. What are kidney stones going to be doing? They're going to be tearing up some tissue, right? These stones are not smooth little, stones. They're ragged. They're kind of crystal-like, and so they're going to be damaging and causing some passage of blood. Maybe not because the glomerulus is damaged, but because it's irritating the mucosal layer. We looked at specific gravity, right? And we've all got a sense of that. And specific gravity would be an indication of how concentrated your urine is. And that would be in osmoles. And we haven't talked about osmolarity units. The pH of urine, again, you'll see different numbers, different ranges. I think in the lab manual it says 3 to 7 is normal. Here I'm saying 4.5 to 8.2 is normal. Okay, so it's pretty variable. I'm not going to ask you what's the normal range of urine because there's different sources of this. But typically your urine is going to be slightly acidic, right? It's usually going to be on average in that 6-ish range. Chemically, it's mostly water, right? Your urine is mostly water with about 5% solutes, and those solutes would include, urea and sodium chloride and creatinine and uric acid and all those things we talked about getting rid of. Plus, you might have a little bit of bilirubin. It is abnormal to find glucose. It is abnormal to find free hemoglobin, albumin, ketones, or bile in your urine. If those are there, they're indications of what we looked at on the chem strip, right? If you have glucose, you would have glucoseuria and perhaps diabetes. If you had albumin, albuminuria, or proteinuria, you would have perhaps some kidney damage if you were always putting lots of protein into your urine. Ketones. Ketones along with glucose would be a really good sign that you're dealing with a diabetic patient. Bile pigments would suggest there's something going on with the liver. Volume, 1 to 2 liters a day. If you have polyuria, you're going more than that. So excessive much urination is polyuria. Oliguria, what does oligo mean? Oligodendrocytes were cells that reached out with a limited, deficient, or few number of arms to myelinate neurons of the central nervous system. So oliguria would be deficient or low output of urine. Less than 500 mils a day. We're a little bit worried. You may remember that we talked in the very first lecture that 500 milliliters a day was your obligatory water loss. You have to lose 500 mils a day to get rid of your waste products. And if you are not releasing at least half a liter a day, then your toxic levels are probably increasing, right? So that obligatory water loss, what you must lose every day. And then if you are not losing any urine, anuria, right? You pretty much have no urine output, which suggests that you're just amazingly dehydrated. You've got shock, right? Remember, all shock is a low blood pressure, and your pressure is so low that your kidneys have shut down. Or you've got a kidney disease where your kidneys aren't working, or maybe you've got an enlarged prostate. That's not an issue with the kidneys, that's an issue with blockage, right? The prostate's so enlarged that urine is not passing. Again, under 400 mils, under 500 mils a day, somewhere in that range, you're in trouble because you're not getting rid of your waste products. Diabetes, I'll mention briefly a little bit about diabetes because that's what we've been focusing on the most as far as seeing abnormal things in the urine. Diabetes comes in at least four types: type 1, type 2, gestational, and insipidus. The first three have to do with glucose. The last one has nothing to do with sugar. And diabetes, dia meaning what? Through, right? So in diabetes we have something passing through. In this case, there's a high concentration of glucose. And why would glucoseuria is a sign of diabetes, but why would polyuria also be part of diabetes? Polyuria, excessive urination. That's one of the signs of diabetes. Why?
Diabetics are more thirsty.
They are more thirsty. They're more thirsty because they're urinating more. And why are they urinating more? Because they have sugar in their urine. Solutes suck. So the more you have of any kind of solute in your urine, water is going to be drawn out of the body. Right? So because there's sugar being lost, water will be drawn with that sugar. So a diabetic is losing more water than a normal person. So of course they're thirsty, right? So we say diabetics are thirsty. Why? Because they are urinating more because diabetics have polyuria. So polydipsia, excessive thirst, polyuria, and glucoseuria are all signs of diabetes, right? Excessive thirst, excessive urination, and glucose in the urine. Now, the last type in diabetes insipidus, this has nothing to do with glucose at all. Diabetes insipidus, DI, is a problem with the low secretion of ADH. If my body couldn't make ADH, then my body could not regulate what? Urine output. People with diabetes insipidus are passing more urine or passing more water than they can because ADH can't stop it. There's no way for the body to stop that. And people with DI will be urinating 30 liters a day sometimes, right? They constantly are urinating. They're constantly drinking, and they're constantly urinating. Medications that can cause you to release more urine collectively are called the diuretics, right? Diuresis. So diuretics are going to cause you to increase your GFR, maybe. So let's take a look. We talked about caffeine in the past, in passing in 105. So caffeine's a diuretic because it increases the GFR, and it increases the GFR by dilating the afferent arteriole. More is going in. You could also, though, reduce reabsorption because alcohol is a diuretic. Alcohol inhibits ADH. Or there could be what's called loop diuretics, a whole class of medications called loop diuretics. And what they're doing is they're interfering with that countercurrent multiplier. If you're interfering with the countercurrent multiplier, you're reducing the saltiness of the medulla. If you're reducing the saltiness of the medulla, then your urine would be less concentrated, right? So you're going to be peeing more. So loop diuretics, so there's 3 different ways we can do this. If a person has some significant high blood pressure issues, they may be given one type of diuretic. And then if that isn't sufficient, they may be given a second class of diuretics because it works on a different part of the kidney, but they would collectively do more to get rid of fluid off the body. If you have congestive heart failure, you've got fluid building up in the lungs. Again, those patients would be given lots of diuretics to get rid of that fluid. So just like last week in lab, we did sort of pulmonary function tests with the spirometer, and we looked at FEV1 and FVC. We can also do RFTs, renal function tests, and in doing this, we're checking for kidney disease, seeing how bad it is, monitoring progress, all the things that we can do, and basically we did that, right? We did a urinalysis. That's sort of a renal function test. There's a lot more. There can be a lot more fancy ones to it. We can also measure your renal clearance and also measure your GFR if necessary. I'm not going to spend a lot of time going through all this detail, but renal clearance is basically defined as the volume of plasma of a particular waste that is completely removed in 1 minute. In other words, some medications are cleared more quickly through your kidneys. Others are cleared more slowly. So if you have a high clearance, a medication that has a high renal clearance, it would wash that particular product or waste product out of your body very, very quickly. What I want you to see is that your net clearance is influenced by these 3 things. This is what I definitely want you to focus here for a moment. Do you agree that the ability of your kidneys to get rid of something or to have something show up in the urine is connected to your GFR, your glomerular filtration? It had to get filtered. And then secretion. What is secretion? Adding even more to it, to the tube, minus the amount that's reabsorbed. Think about it. If I'm a molecule floating through your blood and I'm going to end up in your urine, what's going to influence that? How quickly I'm filtered through the kidney, how much of me would be secreted, added even more to the tube, minus however much is reabsorbed. Collectively, those 3 things would equal the renal clearance. And I think I may do a little jump here. Let me just take a look. Yeah, I'm not going to go through the math here. So I'm not going to work us through the math here. But you can determine renal clearance in the clinic by comparing it to other molecules. Typically, your clearance is going to be compared to GFR. So to normal GFR, just look at this number. Your normal GFR is 125 milliliters per minute. Normal GFR. So what does that mean? All of your nephrons collectively are creating 125 milliliters of filtrate per minute. And if you multiply that by a day, what do we get? That's where you get the 180 liters for guys, right? Or 150 liters for women. So you're always going to compare your clearance with GFR because GFR is sort of the baseline. What would kidney disease do? Kidney disease would do what? Probably if you have fewer glomerulus or glomeruli, right? What would you do? You would have a problem with, right? You'd have lower GFR. And if you have lower GFR, that means you wouldn't be filtering as much or you wouldn't be clearing as much, would you? So if you have kidney failure, it might affect your medications. Does that make sense? If you have kidney failure, you don't need to be taking medications as often because you're not filtering as much. So you'd actually have a buildup of medication. So a doctor has to be really careful. If a patient's having renal failure or renal issues, they got to watch their doses really carefully because they're not clearing as much, they're not filtering as much, so they could certainly overdose them, if you will, or give them a higher level of medication than what is necessary. I'm not going to worry about the inulin or the creatinine, okay? So I'm not going to worry about that. Now I got to make sure it's not in the final, but I'm not going to worry about it, okay? Now, the next 10 slides or so are about the bladder. And this is just about the ureters, the bladder, and the urethra, and the comparing and contrasting of the male and female urethra. And that's 105 stuff. So I'm just going to flip through that, and you can refer to just reading through this about the bladder, about the layers of the bladder, about the capacity of the bladder. I mean, a moderately full bladder is about 500 mils. About the most the average person can handle is up to about a liter, but typically 700 to 800 mils. And I'll just say this really quick. What do we have here in the bladder? Because there were some structures like this on your lab list as well. So coming down to the bladder, what do we have here? These are the ureters. And inside, this is where it would open up, right? The ureters dump into the bladder here, and there's an imaginary triangular structure called the trigone. And it's the two openings for the ureter and the opening for the urethra. At the beginning of this, right in here, what muscle do I find? The internal urethral sphincter. That is involuntary muscle, right? Smooth muscle. And then a little further down in the layer of the urogenital diaphragm is another muscle sphincter that is the external sphincter. That is voluntary muscle. This is a female bladder. If this were male, I would see what right here? Prostate gland, right? That would be about the only difference. And then, of course, a little bit more length here. You can have the building up of kidney stones. And you've heard me say the word calculus before. Remember, calculus is hard, right? Calculus, math is hard. Calculus is hard, stone-like material. So calculi, hard. And what is kidney stones? Typically, they're calcium phosphate. And I remember saying to you long ago that you never want calcium and phosphate in high concentrations in the same place. Because when calcium and phosphate are together in high concentrations, they make stones. So you never want to have calcium and phosphate coming together in high concentrations. Usually they're yin-yang. Usually if there's high calcium, there's low phosphate, and vice versa. Kidney stones are quite painful, so I've been told. Anybody ever experienced a kidney stone? You want to share the experience? I've heard it's excruciating. What would cause kidney stones? There was a few things. Gee, my doctor said I should take calcium because my grandmother had osteoporosis. So if a little bit of calcium is good, more must be better. No. Right? If you get those chocolate or caramel flavored calcium chews, don't treat them like candy. Right? Don't eat 4 and 5 of those things a day. Oh, I'm hungry, I'll have a calcium chew. Right? Because too much calcium will build up the potential of kidney stones. Dehydrated? Why? If you're not flushing your kidneys, then you already saw in lab those crystals that are forming in your urine. And if you're not flushing the kidneys enough, those little crystals can get bigger, right? Kidney stones. pH imbalances. Some of the kidney stones or some of the stones are more likely to occur when there's a higher or lower abnormal pH. If you have frequent urinary tract infections, again, you're not pushing and flushing urine as often as you should. And an increased prostate gland, guys, would do what? Keep urine in the bladder longer than it would normally be there. And again, that could start leading or bladdering up further into the kidney. And that could lead to the up regulation or the up number of kidney stones. So we're going to put you in a lithotripsor, which is basically a sonicator type thing that will hopefully pulverize the stones, and you can pass them naturally. That's what's usually done as a non-surgical solution. Okay. And then the urethra in the male has the three parts. There is the prostate gland sitting here, of course. There's the internal sphincter, the external sphincter, and the part going through the prostate is the prostatic urethra. The part going through the urogenital diaphragm is the membranous urethra, and then the majority of it is the penile or the spongy urethra. UTIs, cystitis, infection of the bladder. You'll see that word in the lab when we're talking about what's going on with pHs and things. You'll see the word cystitis. Pyelitis, infection of the pelvis. Ooh, if you've got infection going all the way to the pelvis, that doesn't sound good, does it? Okay? And pyelonephritis, infection going all the way up into the cortex. So these infections start from the urethra, right, and go up toward the kidney. So an infection of the urethra up into the bladder, that would be a UTI. But if it really starts to affect the bladder, now we're talking cystitis. If it's going up into the renal pelvis, pyelitis. If it's going all the way up into the cortex, now you're talking pyelonephritis. Now what can go wrong there? Those bacteria, they're up where? They're up in the nephron. So now where are they? They're just a short step from getting into your bloodstream, right? So a kidney infection that is untreated, that gets worse, that bacteria can go all the way up into, pass, if you will, backwards through the glomerulus and start infecting your blood and create a systemic infection. When we're voiding urine, there's a detrusor muscle. The detrusor muscle is in the wall of the bladder. And so that's going to relax. And then there are the sphincters that are going to open up under control. Micturition is the act of urinating, and there's a reflex involved here. Part of it is involuntary. So there are stretch receptors that detect how full your bladder is. And they're going to send signals to your spinal cord because this, again, is a reflex. Remember, the spinal cord is in charge of reflexes. So the signal goes to your spinal cord, and then that's going to come down from the spinal cord to your bladder, to your to your sacral nerves, S2 and S3. That makes sense, right? At the very bottom of your sacrum, those spinal nerves are going to go off to your bladder. And that's going to then cause your detrusor muscle and your internal sphincters to involuntarily begin to relax. And then finally, hopefully we have control over this, right? Finally we get to the voluntary control where we now are able to, from our conscious parts of our brain, now send signals down for urination. So it starts off as involuntary and then goes to voluntary. Let's look at the pathway here. The 8 steps are listed over here on the right. But number 1, what's going on? Your bladder's filling. There are stretch receptors in the wall of your bladder. They're going to send signals just to your spinal cord, basically. And those signals right here are going to come back, and they're going to inform S2 and S3, right? S2 and S3. And they're going to send signals to your detrusor muscle and to the internal sphincter. There's also going to be signals, though, that are going to go up to your consciousness and then come back down. And those are going to be voluntary signals that are then going to travel through the pudendal nerve. You may have seen the pudendal nerve. I think it was on our list. Pudendal nerve is the nerve going to the bladder, and it's going to now relax the external sphincter. If your bladder is not full enough to trigger their normal micturition reflex, and maybe some of you experienced that in lab this week, you're like, oh, I don't really have to go. Well, go anyway. Well, how does that work? You're going to have to squeeze harder, aren't you, to make it happen. So if you don't have the normal reflex signals going on, then you can get help from the Valsalva maneuver, which is going to help compress the bladder and hopefully get urination flowing again. You know, when you go to the doctor, you say, oh, I just went, I don't have to go. Well, go anyway. It's time for your physical, right? It's going to take some extra effort, Valsalva maneuver to make that occur. This little video goes through this whole micturition reflex. If your kidneys are not sufficiently working, then with medical miracles, we have dialysis, right? So now, if the kidneys aren't functioning to cleanse your blood, you can go through hemodialysis or dialysis. Basically, if you're going to be a long-term patient, they'll put a shunt in your arm or in your neck or wherever they can. Patients who have been long-term dialysis patients, these shunts start to fail, and you'll see shunts now in their neck, hanging out of their neck and other places, where they're constantly going into the clinic 2 and 3 times a week to have their blood drawn out. And the blood is going into a machine, it's filtering it out, if you will, and then returning the blood without the waste products. Typically, you do this 2 to 3 times a week. Right before a patient goes in, they're feeling kind of cruddy because the waste products are accumulating. And so they're feeling kind of cruddy, they need to go in for the dialysis, and then dialysis takes quite a bit out of you, energy-wise. And so after dialysis, patients are kind of weak. So they kind of have, you know, cruddy days and better days, but they get to sustain life pretty well. If you go on dialysis, the last time I heard, it is an automatic full-time disability status. So it's really hard to carry on a job if 3 days a week, 3 mornings a week, or 3 afternoons a week you're in the clinic. So I think it's an automatic full-time disability status unless you choose to try to work around it. So what's going to cause you to have renal insufficiency, renal damage to the point where you need dialysis? It could come from a lifetime of hypertension, right? Hypertension is going to do what? What's hypertension going to do? High pressure into the kidneys is going to slowly destroy your glomeruli, and no longer can they filter. Chronic kidney infections, trauma, extended periods of hypoxia where the kidneys are not being fed, heavy metals, all of those things can lead to problems with the nephron destruction. If it's a short-term injury, then the nephrons can regenerate. So if a person has a major infection or damage to the kidneys, a fair amount of the nephrons can regenerate and come back. But if there's any kind of long-term issues, they do not regenerate. You can survive with 1/3 of one kidney. And I think I mentioned this the other day, or did I? I think I did, right? So 1/3 of one kidney, what is that? 1/6 of function, about 16%. So you can survive on 15, 16, 20% of function with no issues. It's not until you have lower than that you're going to start having problems. The problem is by the time you are down to 16% kidney function, we can't restore it. So at that point, all we can do is try to maintain what you have left and try to keep you off dialysis as soon as possible, as long as possible. Okay, and that brings us to the end of that, right? So let's take just a couple minute break. I'm going to come back and we're going to talk a little bit about the acid-base regulation, and then I'll be re-recording that first part for you so it's all one big presentation. So we're coming full circle. The very first chapter we covered in day 1 was chapter 24. And you may recall we talked about water balance and ADH and osmoreceptors and the hypothalamus. And that set us up to talk about sodium and potassium, because we had to understand that sodium was always the electrolyte in the extracellular fluid and that potassium was always the electrolyte in the intracellular fluid. And we had to understand that story to move on to the nervous system to talk about resting membrane potentials and action potentials. Okay? So that's where we started the whole course, going into the nervous system and action potentials. We're not coming back to look necessarily at water and electrolytes, although we've been doing that right now. The thing I did not cover with you last time, though, was about acid and base. We did talk about water. We did talk a little bit about sodium and potassium way back when. So I'm going to have you read through these first few slides and just remind yourself of what we've already been talking about, and that is that the body is having to keep your fluid levels, your electrolyte balance, and your acid-base balance in check, and that your urinary system is a large part of this, but it's not alone, right? You've got a lot of other systems that are also contributing to this balance. So sodium, don't worry about the numbers, but I've already mentioned aldosterone, right? I've already mentioned aldosterone, and I've already mentioned the renin-angiotensin-aldosterone mechanism. So you already know what's on these slides. This is a really nice picture, though. What happens, I told you that aldosterone was released, or did what? Aldosterone causes the body to do what? Reabsorb more water and more sodium. So what would trigger aldosterone? Besides renin. But what else would trigger aldosterone? There must not be enough sodium, right? You're going to reabsorb more sodium as a result. So if there's not enough sodium, hyponatremia. Natrium is sodium. So hyponatremia would be what? Low sodium levels of the blood. And if you have hyponatremia, you're going to stimulate the adrenal cortex to make more aldosterone. We've already talked about renin, right? So I told you earlier, either low sodium or high potassium, either one, would cause your body to make more aldosterone, and then that's going to cause your body to reabsorb more sodium. We've already talked about ADH. We've already talked about atrionatriuretic peptide. So moving on. Oh, I should go back, though. This is kind of fun. I know all of you guys need to know this. Estrogen mimics aldosterone. So, when is estrogen higher levels? Anybody remember? It is during pregnancy, but also estrogen does a cycle up right before ovulation, right? So this is why, ladies, retaining fluid. Okay, so estrogen mimics aldosterone chemically and causes the bloating and all those lovely effects that you experience. Okay, so I just wanted to point out that the sex hormones do have an effect on water regulation and sodium regulation, and that's where the connection is. It's rather rare that we have hyper or hyponatremia because our body is so fundamentally connected to it. I mean, it's really hard to get your salt levels high or low. It could happen, but it doesn't happen too often because it's so critical to all functions. Potassium, we've already talked about potassium before being the most prominent intracellular electrolyte. Moving on. Well, I should say this. What if you had hyperkalemia? Hyperkalemia. What would be the side effects? We talked about this long ago, but what would happen with hyperkalemia? Too much potassium. I mean, this is a great way to kill off somebody you don't like, right? So if you give somebody a big old bolus of potassium, you can put them in cardiac arrest because you're going to throw off their sodium potassium levels and the ability of the heart to contract, the ability of your muscles to contract, and it also screws around with the action potentials. So just think about what would happen if your sodium was high or your sodium was low, your potassium, those kinds of things. Potassium is a far more dangerous imbalance than sodium. Right? Sodium rarely gets too out of kilter. Potassium has far greater implications. You may recall I told you that potassium was really the main electrolyte maintaining your resting membrane potential. So potassium is very, very important. Now, let's get over here to pass by chloride and calcium. We've already talked about PTH today and phosphates. Now, what I want to get over to is acid-base. This is the focus for this. So acid-base, we know acids are things with a pH less than 7. An acid would be something that when put into water gives up a hydrogen. Strong acids give off a lot of hydrogen. Weak acids don't give off as much. Bases, turn it around. Bases are chemicals that can accept that H+. In 105, I talked about how bases were things that put off OH-. But another way of thinking about it is that they can accept H+. So we know that, and we've been talking about how the kidneys are helping to maintain your acid-base levels. Why do we have to keep the blood pH at 7.4? I've said over and over, it's very important, right? You have lots and lots of enzymes in your blood. And those enzymes work ideally at 7.4. And if the pH changes higher or lower, then those enzymes can very quickly become denatured. That is, they don't work. And once they're denatured, they don't spring back into active form. And so once you destroy those enzymes, they are not available for you. And those slight changes can have dramatic effects on your entire metabolic pathway. So your whole metabolism, if you will, can shut down if your pH is vastly off from 7.4. 735 to 745, that's your normal range. 735 to 745. Anything beyond that, we've got some issues. Now, where is all this acid coming from? We started off this whole conversation with the urinary system saying that your body is producing acids all the time as part of your metabolism. You're making all the time lactic acid. If you have any kind of anaerobic fermentation going on in your muscles, right, you've got lactic acid being formed. You've got phosphoric acid always being produced as you break down your nucleic acids. You're making fatty acids and ketones as you break down your fats. And finally, there's carbonic acid, as you know, is being formed from your CO2. So your body's always becoming more acidic. There's no way to avoid it. Your normal metabolism is driving your pH down. So we've got to get rid of that acid. And we have to have a way of maintaining your pH levels. And if you have a hot tub or a pool, you know that you throw sodium bicarb, you throw bicarbonate, baking soda into your pool or hot tub to keep the pH well buffered, and your blood does the same thing. Your blood has a fair amount of bicarbonate in it. And a bicarb is a buffer, and what that's going to do is keep the pH from moving. So even though your body's making acid, your pH of your blood is going to stay relatively constant. It's not going to allow the pH to change dramatically. Your urinary system does a lot to buffer your pH, and your respiratory system also is largely involved. Let's think about this. How is the bicarb is a very common chemical buffer in your blood. There are also phosphates, and some of your proteins also can help to neutralize the pH changes. I'm not going to get into the equations here, but there's one equation here you have seen many times, and that is the middle one. Where have you seen this equation before? Right? CO2 plus water makes H2CO3, that's carbonic acid, plus, or that also then becomes bicarb. HCO3 minus is bicarbonate, and H plus. And we talked about how your CO2 is traveling through your blood largely as carbonic acid, right? That CO2 is carried on hemoglobin, but it's also largely dissolved in and carried through your blood, as this equation tells us about. And it is a reversible reaction. So if you have too much CO2, what's your body going to make? More acid. So if you have too much CO2 in your body, you are going to become more acidic. You're going to have more acid to deal with in your body. Again, your lungs and your kidneys are working together to constantly remove this CO2. And we got that, right? We're always blowing off CO2, and a lot of this bicarb stuff is dissolved in our plasma and is being released in our urine. Our urine's acidic, right? So we're getting rid of acid through our urine, and we're getting rid of CO2 through our lungs. Don't make this harder than it is. Just know you're always blowing off CO2, and your urine is usually acidic. What else do I want to tell you about? Oh, phosphates are also helping to do this. If you haven't had chemistry or you're not a big fan of chemistry, I'm not going to get into all of this. Just know that your phosphates are also helping to buffer your pH, as are many proteins in your body. Okay, many proteins. So we'll just say proteins, phosphates, and bicarb. 3 very important buffering compounds. Now, let's talk about how the respiratory system, we've already been talking about the respiratory system, we know a lot about this. and I think we already know this. We know that the addition of more CO2 in your body is going to do what? Raise your acid levels and lower your pH. I sense from this week's quiz, still too many people screwing up pH and acidity. Too many people did not recall, well, I think they know that when you blow CO2 into a straw, into a beaker, that it becomes more acidic. And then when I asked what happens to the pH, many people in their quiz this week said the pH went up, right? There's still too many people who have this thing flipped. Okay, and I don't know if you're flipping, remembering what happened, or not knowing what happened, or not understanding that more acidity is a lower pH. Okay, we got to not get that one confused. So if you have more CO2 accumulating in your body, you're going to be more acidic, right? And if you remove extra CO2, it's going to be the opposite. How would you be removing too much CO2? What happens that you can blow off more CO2 than what your body's making? Right, if you're hyperventilating, you're blowing off more CO2 than your body needs to, so your pH would not go down, but instead your pH would go up. Okay, been there. We know that the respiratory system is involved with this. That's exactly what this equation is telling me. That with more CO2, right, you're always making CO2 as a part of your metabolism, and CO2 is being blown off by your lungs. More CO2 lowers pH. If you blow off more CO2, you're going to raise your pH, as we just said. The kidneys are also helping to control the pH. Your kidneys are secreting hydrogen. What does that word mean again? Secreting. Taking acid from the blood and putting it into the tubule. Concentrating the acid into the urine. So just to look at that picture here, what this means is that there is CO2. I've got it marked here as CO2. There is CO2 in your blood. Now, you know that when you see CO2 in the blood, we're talking about, quote, things that are acidic. So here we are in the tubule. Sorry, we're in the capillaries, and we've got CO2. And that CO2 is going to be transferred through the cells and do what? Go into the tubule. That's the idea of secretion, right? We're going from the blood into the tube, and as a result, the urine would be more acidic. I'm not going to worry about this slide. So cross off this slide, and the next one, and the next one. Okay? I want to get to a couple of ideas in the last couple of slides. It's a balancing act, isn't it? So we've got a normal blood pH of 7.4. If your blood pH is over 7.45, we're going to say that you have alkalosis. If the pH is less than 7.35, we're going to say you have acidosis. You're dead at 8, you're dead at 6.8. Right? So we don't have a whole lot of variation that's available here. Again, your kidneys are balancing this out along with your respiratory system. Here's what I want to get to. What happens if you become acidotic? What happens if the pH of your blood goes below 7.35? What do you think? That means that there's too much acid, so that acid is going to diffuse into your cells. Oh, by the way, when acid comes in, it pushes out potassium. So if you don't have enough potassium, that's going to cause your cell membranes to become hyperpolarized. Right? What does that mean? Down lower. That means it's going to make it more difficult to reach threshold. Oh my goodness. You're going to make me think about action potentials again, aren't you, Mac? Okay? But we're hyperpolarized. We have less potassium. It's going to make it harder to reach threshold. So what's going to happen? Your CNS, your central nervous system, will become depressed, right? Not as able to fire off your action potentials. And you can become disoriented, confused, even comatose, death. So as you move down more and more acidic, it leads to a depression, a shutdown of the central nervous system. If instead you have alkalosis, that means now that you've got too much hydrogen coming out of your cells, and potassium will do the same thing. The membranes will now depolarize. What's going to happen now? They're going to be overstimulated, right? It's going to be too easy to reach threshold. Right, it's going to be too easy to reach threshold. Okay, it's going to get too easy to reach threshold. And it looks like I've got one of those 610 shutdowns on the internet. We'll see what happens here. It's not one of the best days for this, is it? Okay, so how are we going to deal with this? If we are alkalosis, we're going to become too hyper, right? Our nervous system is going to become hyper or too overstimulated. That's going to lead to muscle spasms. It's going to lead to convulsions, respiratory, paralysis. Now, what's the body going to do about this? Okay, we're going to have two different kinds of acid-base imbalances in our body, respiratory and metabolic. If I'm telling you that you have a respiratory imbalance, the reason you have the imbalance is because your lungs aren't working. If instead you have a metabolic disorder, it's because your kidneys aren't doing their job. Okay, so when I say respiratory acidosis, it means that you are acidic because your respiratory system is not doing what? Right. If you have respiratory acidosis, it means that your lungs are not adequately blowing off CO2. You're accumulating CO2. If you have respiratory alkalosis, hyperventilation. If you had metabolic acidosis, your kidneys are not sufficiently getting rid of the acid. If you had metabolic alkalosis, then they are what? For some reason, getting rid of too much acid, and the balance is going the other way. So think about those different types of imbalances. Different types of imbalances. And it's all laid out here for you, what your body would do. Now, respiratory acidosis would occur, for example, just to get an idea of this, in emphysema. What's emphysema do again? Alveoli get blown out, less gas exchange, CO2 is accumulating, you can't get it out, the body becomes more acidic. The respiratory system can't deal with it, right? It's failing. So what system has to come to the rescue? The kidneys, okay? Versus if your kidneys are failing and you're in some sort of metabolic acidosis or alkalosis, obviously the kidneys aren't keeping up on their work. They're now going to depend more on the lungs to pick up the difference, okay? So that's really what this last little bit is focusing on. Okay, so I will fill in those last little gaps. Look at this last little bit on how the body compensates, how it corrects for these acid-base imbalances. And do not go on to fluid placement therapy. So I won't ask you about the fluid, I'm not going to ask you about the fluid replacement therapy. Just stop with the acid and base imbalance. Read through that, and if you have any particular questions on that, please check with me next Tuesday. Now at this point, what can you do? Study. You now have all that you would ever need to do your Connect assignments for acid base and urinary. You also can do the first quiz. There are 2 quizzes out there on urinary. And I would definitely get those done this weekend so that you don't have to fuss too much on the reproductive after that. I know you've got your lab exam, but that's on urinary too. So as you're focusing on the urinary system this weekend, get that Connect quiz done, get the Connect assignments done, and don't forget also to do the APR quiz, which is also respiratory and urinary. So you're all in the same family here as far as what you're thinking about this weekend. Enjoy, and I'll see you all very, very soon.